Hi everyone, welcome to chapter 21, the immune system. Now remember, the immune system is the second portion of chapter 21. There is also the lymphatic system, which is the first portion of the chapter, but that was self-study. Now, before watching this lecture and diving into immune, it is strongly recommended that you have already done the self-study lymphatic portion of this chapter, because we are going to talk about stuff in these lectures that depend on you knowing material from the self-study lymphatic. So if you have not already done the lymphatic material, please go back and do that and then come back to this lecture and the following lectures. Now, when we talk about the immune system, it's not really an organ system the way all of the other systems that we've talked about so far in Bio 137 and in Bio 139. The immune system is actually a functional system. That means we're not going to point to an organ and say this organ is part of the immune system. No, the immune system is just a bunch of cells and tissues that work together to keep us healthy, to fight off pathogens. And the immune system is broken down into two separate portions. The innate side, which is what we're going to discuss today, is non-specific. It is indiscriminate with what it will fight off. And the next two lectures will focus on the adaptive portion, and this is very specific. So innate just says, this looks like something that could potentially make me sick. We're going to get rid of it. Adaptive says, this looks like flu type H1N1 of a certain strain. And it will make sure that we don't get sick from that. So they function very, very differently. Now, diving into innate defenses, the innate side, the non-specific side of our immune system, it's concerned with preventing entry and spread of pathogens. And again, pathogens are just something that could potentially make us sick, do us harm. And the innate defenses are further broken down into two different portions. The first line of defense is what will prevent entry of a pathogen. The second line of defense is if something has passed that first line, now we're going to prevent it from spreading, from reproducing, and from moving throughout our body. So, first line of defense, these are barriers. Just something to prevent pathogens from getting into our body in the first place. Things like our skin, mucous membranes, and some other things that we'll talk about. The second line of defense is there to prevent spread of pathogens if they do make it into our body. And it will contain various proteins, phagocytes, and other cell types. Now, the innate defenses are very local. They focus on something in a specific part of our body. They are non-specific. As I said, they are just there to prevent anything that could potentially make us sick. And they lack memory. So that will make more sense as we get into the next two lectures. But memory is just something that our immune system has in the adaptive side. That's if something shows up and it makes us sick. If it shows up again, we already have an arsenal in place to fight it. It will not make us sick a second time. Now, the innate defenses, every time something shows up, whether it's the first time or the 40th time, we treat it as though it is the first time. There is no memory of the previous encounter with a specific pathogen on the innate side of our defenses. So that's just kind of an overview, but now let's start to actually talk some specifics. And we're going to look at, like I said, today, the innate side, the non-specific side. And we're going to begin with the first line of defenses, the surface barriers. Now, the first line contains things like the skin. Now, this is a physical barrier. It's kind of like a wall between the inside of our body and the outside of our body. And like we said in the not really attendance questions, keratin is what gives our skin that toughness, that waterproofing ability. But it's also going to be very dry. It's inhospitable. Things don't want to grow in those types of conditions. Now, our skin is actually covered completely in bacteria. But those are bacteria that are not harmful to us. They are our own bacteria. Everybody has their own native bacteria. But if something shows up that your body is not used to, then it's not going to have a very easy time colonizing your skin because the keratin creates a very inhospitable environment. There are also various chemical barriers. We have something called the acid mantle. Now, our skin itself has a very low pH, but also inside of our mouth, inside of our nose, inside the anus, inside the vagina, all of those areas have a very low pH. And this is because of things like sweat, oil secretions, and various other secretions that all lower the pH. And low pH is antimicrobial. Things do not like to grow in an acidic environment. Dermacidin. Dermacidin is a component of sweat. So when we sweat, dermacidin is spread across our skin, and dermacidin is also antibacterial. Defensins. Defensins is a collective term for various antimicrobial proteins, and they're released from several different leukocytes. Now, leukocytes typically we think of in our bloodstream, but as they move through the skin, they also release these defensins. And defensins, while they are functional on their own, they also are enhanced by vitamin D, which is why people will say, if you feel bad, get some sunlight, because sunlight activates vitamin D, and vitamin D makes defensins more potent. Mucus. Mucus is a protein released from various cells throughout our body, such as in our respiratory system, in the trachea, inside the nasal cavity, deeper down into the lungs, but also other places throughout our body. And mucus, the protein, when it gets mixed with water, when it comes into contact with water, it produces that slimy mucus that we tend to think of. And this will trap different pathogens, it will trap debris. And in our respiratory system, for example, once that mucus has all that debris trapped in it, the cilia are constantly moving that mucus up so that we could cough it out. Lysozyme. Lysozyme is an enzyme in places like inside our nose, inside our mouth, in our tears. And lysozyme is an enzyme that dissolves cell walls. Bacteria have cell walls. And if we dissolve those, then the cell itself will die. And hyaluronic acid. Hyaluronic acid is a component of areolar connective tissue, and remember, that's found just beneath the epidermis. It's found just beneath the tissues of most of our bodies. Now, this areolar tissue and its hyaluronic acid is very thick and viscous, and it's very difficult for pathogens to cross through that hyaluronic acid. But let's say something does get past that first line of defense. It's now entered our body. So our immune system's second line of defense is going to be concerned with prevention of spread. We don't want those bacteria or whatever it happens to be. I will say bacteria a lot, but this is going to be bacteria, viruses, parasitic worms, certain fungi, lots of different things are pathogens. Now, we want to make sure that they can't reproduce. We want to make sure that they can't spread throughout our body. So first, let's look at various leukocytes and macrophages, which again are those monocytes that have left the bloodstream and gone through a bit of a change. These cells are going to have something called pattern recognition receptors. Now, remember, on the surface of every cell in your body, there are certain proteins and carbohydrates called antigens. We talked about some specific ones when we were looking at blood typing and erythrocytes. But every cell in your body will have antigens on their surface. And every living thing will have antigens on its surface. And no two living things, unless they are clones or identical twins, will have antigens that are shaped the same way. Your immune system is going to use antigens. Now, the innate side is going to use antigens one way, but when we talk about adaptive, we're going to go much deeper with how the antigens are used. Now, antigens, even though all living things will have antigens that are shaped differently, harmful bacteria and similar things like we mentioned earlier will have antigens that have similarities. Now, they will all be shaped different, but there will be similarities among harmful bacteria pathogens. Harmful bacteria antigens. There will be similarities between harmful fungi antigens. And these similarities, these patterns, are what these innate second-line defenses are looking for. They have receptors on their cell surface, these leukocytes and macrophages. And these receptors are constantly looking for these patterns. So the way your immune system works is these cells, these parts of your immune system, are constantly bumping up against every surface of your body. Every cell in your body, both your own and anything that might have made its way in. And these pattern recognition receptors on your immune system cells are checking to see what shape those antigens are. And when it comes up on one of these patterns, these shapes that are common among harmful bacteria, that sets off an alarm bell. That basically says, hey, this thing doesn't belong here. So let's see how some of these cells will react if something triggers their pattern recognition receptors. Neutrophils, we've talked about how back in the blood chapter, we saw how neutrophils will phagocytize bacteria. But they do more than just phagocytize. They're capable of creating something called a respiratory burst. And when neutrophils use their pattern recognition receptors and detect something that does not belong there, again, it doesn't know what it is. It just knows that it has a certain shaped antigen that looks out of place. And one of the things that it starts to do is absorbing oxygen from its environment. And it converts this oxygen into 3 or 4 different types of molecules that are very toxic. And it starts to release these new molecules in kind of a cloud around itself. This is called the killing zone. And remember what we said about oxygen? It's really harmful. Well, it's being made even more harmful in these new molecules that are being created. And this killing zone is going to start to kill off the bacteria that are in the area. Now, unfortunately, this also kills the neutrophil, but that's a sacrifice that our immune system is willing to make. All of these bacteria that set off the pattern recognition receptors die around this neutrophil in that killing zone. Now, sometimes this can get a little carried away, and the neutrophils can start having these respiratory bursts, either when they're not needed or if there is a big enough infection. And this can actually lead to breaking down certain things like some of the hyaline cartilage in our joints, and this is what rheumatoid arthritis is. Next, eosinophils. Eosinophils, we talked about how they fight parasitic worms back when we were talking about blood. And eosinophils also are capable of enhancing the action of basophils. Remember, basophils release histamine and heparin and various other chemicals. So eosinophils will aid basophils by making them work more. Lymphocytes. Now, in this chapter, we're going to see that there's various different types of lymphocytes. Lymphocytes, we're going to talk about something called NK cells, or natural killer cells, here in just a little bit. But in the next 2 lectures, we're going to talk about B cells and T cells, which you may have heard of before. These are actually part of your adaptive immune system, so we will not talk about those today. Monocytes, as we keep saying, when they leave the bloodstream and go out into the tissue, they are called macrophages. And that's what we see over here on the right. This big purple cell is a macrophage. These weird extensions are its pseudopods as it's reaching out to either crawl or to grab these bacteria for phagocytosis. But all of those things were brought about because of pattern recognition receptors. Now let's look a little smaller. Now we're going to start to talk about some of the proteins involved in the innate immune system, and we're going to talk about some different types of antimicrobial proteins. First, we're going to talk about something called interferons. And interferons are a class of proteins that are helpful in preventing virus activity. So let's draw how interferons work. Okay, so let's look at how a virus actually works. And what I've drawn here are 2 body cells, and these cells, they could be any cells in our body. It doesn't matter. And so here is a body cell and a nucleus with some DNA inside, and a ribosome. There would be millions of ribosomes. We're just drawing one so that we can illustrate this. And here's another body cell with its nucleus and DNA inside, and a ribosome. But let's draw to see how viruses work. Now, viruses are very, very small, much smaller than what I'm drawing here with regards to the cell. The virus would actually be almost so small that we couldn't see it, even if we drew the cell this big. Now, viruses are weird in that they are not living things. They kind of cross the border between living and non-living. Back in Bio 137, we learned about factors that all living things have. And one of those things is living things have the ability to reproduce, and viruses can't do that. Viruses actually work by tricking their host into reproducing for them. A virus cannot make more copies of a virus. A virus will trick a cell into making copies of the virus. But viruses, and they come in lots of different shapes. They won't all be these round structures, but they all have some sort of outer envelope or capsule with these receptors sticking off from them. And these receptors are very specific. For the most part, a virus that infects a dog will not infect a human. A virus that infects an oak tree will not infect a tomato plant. But they can be even more specific. A virus that infects the type 1 cells in your lungs will not infect a cardiomyocyte. And that's because these little receptors, these spikes sticking off of its surface, will attach to certain proteins on cell surfaces. And when they attach, that's going to trigger some events. So what we see inside here is the genetic material of this virus. And viruses, sometimes they will have DNA, just like we do. Sometimes they will have RNA. Sometimes they'll have both. And there's even variations on that DNA and RNA. Sometimes it's single-stranded DNA. Sometimes it's double-stranded like ours. Sometimes it's single-stranded RNA. Sometimes it's double-stranded. Sometimes it's something called positive sense or negative sense. There's all different variations. So that's another reason why we don't consider them alive, is because they don't always have DNA. Now, when the virus attaches, what it does is it will inject that genetic material. For purposes of this, we're going to say this is RNA. Now, if it was DNA, everything that we're going to draw would still happen. There would just be some extra steps that we don't need to worry about. So we're just going to say that this is an RNA virus. And once a virus attaches and injects its genetic material, the virus itself, that shell, it degrades. It's no longer needed. Because that RNA, it contains all of the genes from that virus. And human RNA, for the most part, looks like dog RNA, chimpanzee RNA, carrot RNA, and virus RNA. They all still are made of those same 4 letters. Remember, A, C, G, and U, adenine, cytosine, guanine, and uracil. Well, virus RNA is also made of those same 4 letters, just arranged in different orders. And what's the job of a ribosome? This little guy right here. Ribosomes, remember, they attach to RNA, they read the RNA, and they make whatever proteins that RNA says to make. So this ribosome is going to attach to this RNA. And when it reads this RNA, it's going to make all of the pieces needed to make a brand new virus. And there we go. Now, meanwhile, this RNA is also going to be copied. So there will be RNA in this new virus. But then it's going to do it again. And it's going to do it again, and it's going to do it again. Cells that are affected by a virus, they kind of become preoccupied. And this cell starts making virus after virus after virus after virus. And it's never going to stop making that virus. Now, it may stop for a little while, but it will start again. And it's just going to keep making this virus. So this cell, once it becomes infected by a virus, it's going to be forever infected by this virus. And there's no fixing it. We will see in one of our upcoming lectures what happens to this cell in the end. But now we're going to see, how do we make sure that this virus doesn't just keep getting worse? Because once these new viruses are made, they leave the cell. And what do? They go to neighboring cells to do the whole thing again. So this cell is so preoccupied with making viruses that it's now not doing a lot of the stuff that it needs to do. Its daily activities start to drop off. And even though there's no fix for this cell, there is something else that comes of it. When this cell's other activities start to drop off, that tells it that, hey, I may be getting infected by a virus. And what it does is it will make a protein called interferon. And interferon does absolutely nothing for this cell. But the interferon is secreted by this cell, and it travels to neighboring cells. And interferon goes to the DNA of neighboring cells and tells it to make antiviral proteins. And these antiviral proteins do several different things. We're going to look at one of the bigger things that it does. It actually tells ribosomes to stop reading RNA for a little bit. Now, what happens is when this virus comes along and injects its RNA, remember, the virus, once it injects its RNA, it degrades. So that virus itself is gone. All that's left is its RNA. And the RNA comes into the cell, is ready to be read by a ribosome, but there's no ribosomes there to read it. Now, let's think about something that naturally happens in our cells. When our cells make our own RNA, the reason that that RNA doesn't trigger proteins to be made forever, over and over and over, is because that ribosome, as it's reading the RNA starts to be degraded. It starts to be eaten away. Even our own RNA starts to be eaten away. So there are enzymes in our cells that get rid of RNA. That viral RNA is no different. It degrades. And eventually, those antiviral proteins stop blocking our ribosomes. But that's okay because there's nothing left of that virus. That virus never infected this cell. So let's see kind of what happened big picture. This cell became infected by a virus. It made interferon to ensure that neighboring cells do not get infected by that same virus. It did absolutely nothing for itself. Once a cell becomes infected by a virus, it will be forever infected by that virus. There is no stopping that. There is no fixing that. But it saved its neighbor by using interferon, and the neighboring cell produced antiviral proteins to ensure that it did not get infected by the virus. Now, one of the examples that I always give to kind of drive this home is, let's say this is someone's house, and in the middle of the night, a burglar comes along and ransacks this house. Meanwhile, the homeowner is barricaded in their bedroom, and they hear that their house is being barricaded, is being ransacked. So what does this person do? Well, surely they'll call the police, but maybe they also call their neighbors, and they tell the neighbors, hey, there's a burglar in the area. He might show up to your house next. Well, this house where the burglar is right now, there's no unransacking it. It has been ransacked. And then when the burglar is done, he leaves and he goes to the neighbor's house. But the neighbor knows. And the neighbor sits there by the front door with a shotgun ready to take care of the burglar when he comes in. This house will not be ransacked. So that's kind of how this works. Interferon is produced by virus-infected cells to warn uninfected cells to make antiviral proteins. The antiviral proteins ensure that the uninfected cell does not become infected. All right. Next, antimicrobial proteins, the complement cascade. So complement proteins are a group of many different proteins in our blood that, for the most part, are circulating doing nothing. This is a trend that we've seen a few times now. Remember, there's lots of stuff in our plasma that most of the time does nothing until it is activated. Well, let's see what happens when these complement proteins are activated. There's lots of different things that can happen. And in your book, it talks about different pathways. You don't need to know the pathways. What I want you to know is, what do they result in? And on page 817, it shows the different pathways and all of the different numbers of complement proteins. You don't need to know any of that. I just want you to know what the complement cascade can result in. So the way the complement proteins work is, one of these proteins recognizes something that doesn't belong in your body, and it sticks to it. And when that protein sticks to it, it changes shape, which causes another one of these proteins to stick to that. And it changes shape, which causes another one of these proteins to stick to that. So they all start sticking together. But depending on which of these complement proteins stick and in which order they stick, we get different outcomes. Maybe they stick together and result in enhanced inflammation. We'll talk about inflammation in just a little bit. Maybe it triggers something called immune clearance. Immune clearance is just the way that our body gets rid of antigen-antibody complexes. When antibodies stick to antigens, that kind of says, hey, this thing doesn't belong here. So things like phagocytes will get rid of those. This just speeds that process up. Opsinization. Opsinization just means phagocytosis is made easier and more likely. Opsinization makes phagocytosis easier and more likely. So if these complement proteins stick to something and trigger opsinization, I always say that it's kind of like if you've ever eaten corn on the cob and you have those little things that you put in the end of it. That's like what opsinization is. Those little things make the corn on the cob easier for you to hold, and easier to eat the corn on the cob. Opsinization, the proteins make it easier for a phagocyte to grab hold of the pathogen, but in addition to that, it also calls phagocytes towards that pathogen. It's kind of like ringing the dinner bell. And lastly, the membrane attack complex, or MAC, which is what we see here. So first, one of these proteins will attach, and then another protein will attach, and another protein will attach, and eventually they all form a ring, and that ring is inserted into the cell wall or the cell membrane of the pathogen. So down here is the inside of the pathogen cell, out here is the outside of the pathogen cell, but there is now a tunnel through. And what happens is water will leak into the cell, contents of the cell will leak out, but eventually the cell will swell up and lyse. It will rupture because of this membrane attack complex. So complement proteins, there's lots of different complement proteins, and when something is out of place in your body, maybe these proteins will start to stick to it. And depending on which proteins stick and which order they stick in, one of these things will happen. All right, let's talk about NK cells, or natural killer cells. Like we said, these are a type of lymphocyte. They're just a specialized lymphocyte. And they kill by inducing apoptosis, and they can also enhance inflammation. So down here we see how they are inducing apoptosis. Remember, apoptosis is programmed cell death, or sometimes it's called cell suicide, because what happens is the cell kills itself. Natural killer cells convince a pathogen cell to kill itself. And this is how it does it. So here is the pathogen cell, the out-of-place cell. When an NK cell uses its pattern recognition receptors to detect something that doesn't belong, the NK cells first release some proteins called perforin. And perforin poke holes in the pathogenic cell. It perforates the cell. And it kind of looks like that membrane attack complex that we just saw. It's different, but it's similar in that it's a tube through the cell. Then the NK cell releases another protein called granzyme. Granzyme leaks in through that hole, and it does a few things. One, it starts to degrade parts of the cell, but more importantly, it triggers apoptosis in this cell. Apoptosis is something that cells do on their own when something goes wrong inside the cell. If something goes wrong inside the cell will recognize that. And to prevent that whatever it was from getting worse and infecting other cells, the cell will kill itself. Well, granzyme tells this cell to go ahead and do some apoptosis. It tells this cell, hey, kill yourself. And the cell does. Its organelles start to break down. Its cytoplasm starts to behave weirdly as different enzymes start to build up inside of it. And once it shrinks down into this useless mass that used to be a cell, a macrophage will come along and eat it. Phagocytosis. So that's how natural killer cells, NK cells, work. In one of our upcoming lectures, we're going to see some other cells that do something very similar to this. Well, let's start talking now about fever. Medically, this is called pyrexia. Fever is something we've all experienced, and we know that it feels absolutely miserable. But fever is actually our body fighting something that is making us sick. The fever, even though it feels miserable, it's a good thing when we're sick. So leukocytes and certain other cells, either damaged cells or macrophages, will release some proteins called pyrogens. And pyrogens circulate in our blood until they get to our hypothalamus. The hypothalamus is what sets our body temperature. It's kind of like our thermostat. And pyrogens tell our hypothalamus to increase that thermostat, to turn the temperature up for just a short term. Now, normally our body temperature is about 98.6 or 37 Celsius. Some of us, it's a little bit lower. Some of us, it's a little bit higher. But it's in that vicinity. 98.6 Fahrenheit is what's considered normal. Now, when these pyrogens are released, the hypothalamus cranks that temperature up, and we get warmer. And that's the big sign of a fever, increased body temperature. But something less obvious goes on during a temperature also. Pyrogens tell the liver and the spleen to sequester iron and zinc. So iron and zinc in our blood and in our tissues, the liver and the spleen start to pull those from circulation. The liver starts to pull iron and zinc out of the blood. The spleen starts to pull iron and zinc out of the blood. And this is because bacteria need iron and zinc in order to reproduce. So what's going on during a fever? Well, bacteria that colonize our body to make us sick, they do that because they like the environment inside of our body. Body temperature is comfortable to them, and that's what they need to make more bacteria. Iron and zinc are needed by the bacteria to make more bacteria. So if our body temperature is raised, well, the bacteria don't like it anymore. First, they can't reproduce, and second, they start to die off because the temperature is too high. If there's no iron and zinc because the liver and spleen have pulled them out of circulation, the bacteria can't reproduce. So fever makes it so that the bacteria can't reproduce, and it starts to kill them off. Now, when we have a fever and we feel miserable, it's really, really tempting to reach for a fever reducer. But that's one of the worst things that you can do, because if you reduce your fever, really all you're doing is rolling out the carpet for those bacteria and saying, here, stay a while, let me make it comfortable for you. Really, one of the better things that you can do when you have a fever, one of the things that I do, is I will sit in a hot bath, just as warm as I can stand it, for as long as I can stand it. I'll keep my temperature up because it will get rid of those bacteria. Now, a couple of catches to that. That's only if your temperature is below 104. Above 104, now you start to run the risk of denaturing your proteins like we talked about before. Remember denaturing proteins at high temperatures? Well, above 104, now it's risky. That's when you want to see a doctor. That's when you want to take some fever reducers. Or if you have small children. When you have small children, definitely listen to their doctor, because small children are much more sensitive to temperature changes, and they can start to have some negative effects even lower than 104. But here is kind of the graph of what happens during a temperature. This is on page 819 of your textbook. So your normal body temperature is always fluctuating. You're always getting a little bit warmer, a little bit cooler, a little bit warmer, a little bit cooler, but typically it's around 98.6 Fahrenheit or 37 Celsius. But when you have fever, you have the onset phase. This is when your body temperature is getting much warmer. And once you get up high enough, now we're going to start to go through something called the stadium phase. This is when we're getting warmer, cooler, warmer, cooler. This is when you go, freezing, burning up, freezing, burning up. And then after it's over and those pyrogens stop being released, now your body temperature comes back to normal. This is called defervescence. So you come back to normal. All right, inflammation. So inflammation is another thing that gets kind of a bad rap. It hurts. It doesn't feel good, but this is our body fighting for itself. Now, inflammation can occur in various parts of our body, and usually when we have inflammation, it's given a name, and that name usually ends in itis. When you hear something that ends in itis, that just means inflammation of whatever. Bronchitis, that's inflammation of your bronchi. And inflammation, we say it has 4 cardinal signs. And those 4 cardinal signs of inflammation are redness, heat, swelling, and pain. Redness, heat, swelling, pain are the 4 cardinal signs of inflammation. And inflammation is triggered by our own body. Specifically, it's typically mast cells. These are cells in our skin that are really similar to basophils. And these mast cells release histamine and a few other chemicals. And that's what triggers the inflammation. And these other chemicals will do a few things. One, they're going to attract our immune system to that area. They're going to attract leukocytes. But they're also going to do a few other things. When we have inflammation, we get vasodilation. Our blood vessels dilate to let more blood come to the area. And they're going to have increased permeability. Remember, our capillaries and venules are really permeable. But they become more permeable when these histamine and other chemicals are being released. But this is very local. This is not across the entire body. Such as up here we see, looks like some inflammation on this little kid's upper arm. But it's very local. It's not so much down here. It's not on his other hand. It's not on his chest. So when we get this vasodilation and the increased permeability locally, we get a few things that happen. First, we get hyperemia. Hyperemia is just excess blood flow. The vasodilation lets more blood come to the area, so we get hyperemia, excess blood flow. And the hyperemia is what causes the redness and the heat. The redness, because there's more blood in this area. We can see it. And the heat is because of that excess blood flow to this area. So this is kind of like a fever, but it's got a different cause, and it's localized. If you were to touch this area, it would be very warm, as opposed to over here, not so much. Exudate. This is the liquid that's leaking out due to this increased permeability. Exudate is similar to plasma. It's liquid, it's got some clotting factors, it's got some antibodies in it. But as that exudate leaks out of the blood vessels into the tissue is going to swell. We get swelling. And swelling is called edema. So as that tissue swells, it's going to press on the nerve endings of that tissue. It's also going to push lymph away. Well, the pushing on the nerve endings is what causes the pain. It makes you aware of that area. But the pushing the lymph away, now if you've read the lymphatic portion of this chapter, you know that that fluid that leaves capillaries is pushed into lymphatic capillaries, is going to travel through lymph vessels. But that fluid, as it moves into the lymph capillaries, any bacteria or anything else that may be in this area right here causing the pain, causing, the reaction, it's going to be carried away along with that fluid in through your lymph system. And when it gets to your lymph nodes, that gives your immune system a chance to say, hey, this is in my body somewhere. I need to prepare for it. And that will be the topic of the next 2 lectures. What happens when things in our lymph nodes are exposed to a pathogen? So here is kind of what's going on during that inflammation process. This is on page 821 of your textbook. So when there is some sort of tissue damage, it looks like somebody stuck a splinter in their hand. Well, that splinter, like everything, is covered in bacteria. So we damage the tissue, we introduce bacteria, and that damaged tissue and even the bacteria themselves are going to start to release some chemicals. And some of those chemicals are going to cause inflammation. So those inflamed chemicals or those inflammatory chemicals are going to trigger leukocytosis. It's going to trigger the red bone marrow to start to make more and more and more leukocytes. The leukocytes exit the bone marrow into the bloodstream, and now we get that vasodilation and the increased permeability. So we have more blood coming to this area, and these inflammatory chemicals, they're like an alarm bell. They're saying, hey, this is where the problem is. So as those white blood cells get to the area where there are more inflammatory chemicals, they start to move to the edge of the blood vessel, and they tumble and crawl along the edge of the blood vessel. That's called margination. Margination is when those white blood cells, those leukocytes, travel to the edge of the blood vessel and they kind of crawl and tumble along the edge of that blood vessel. Now what they're doing is, remember capillaries and venules, remember they are only a single cell layer thick. So where 2 cells come together, there's this little gap. And that gap has been made larger because of that increased permeability. So these margination, these cells are looking for those gaps. And when they find one, they're going to squeeze through, as we see right here. And this is called diapedesis. Diapedesis, those white blood cells are now going to squeeze out of the bloodstream into the tissue. And once they're out into the tissue, remember way back on the 2nd lecture, we talked about common factors among white blood cells. They have something called amoeboid movement and positive chemotaxis. Amoeboid movement allows them to crawl around. Positive chemotaxis means they move toward certain chemicals, such as these inflammatory chemicals. So the white blood cells, they squeeze out with diapedesis. Positive chemotaxis draws them to the area where the problem is, and then we get phagocytosis of the bacteria or whatever it is that happens to be in this area causing problems. Okay, so that is the end of innate immunity. The next 2 lectures are going to be talking about adaptive immunity, the specific immunity that we have so that we can fight off specific pathogens. All right, if you have any questions at all, please let me know, and I will talk to you next time.